Hello and welcome to another biology basics. Now we're going to go through the plasma membrane. So basically the plasma membrane is made up of phospholipid bilayer, and that's represented by these things here. We've got a phosphate head and two fatty acid tails, and they are able to move around each other, hence the fluid, part of the fluid mosaic model. They're in a bilayer. That means there's two. You'll notice that the tails hang out together and the heads are actually on opposite sides. That's because the heads are charged negatively, so they don't like the tails. And the term that we use when we're describing the heads is we say that they are hydrophilic. And if you remember, water also is polar, so has a slightly negative and a slightly positive charge, and so that's why the heads will like water and be hydrophilic, and the tails, which don't have any charge whatsoever because they're basically just made up of hydrogen-carbon chains, repel the water, and so we call them hydrophobic. So what will happen is that these phospholipids will move around each other in this plane, but will not be able to go up and down because the heads and the tails will repel each other. Much in the same way that anything that's outside the cell, which will be up here, unless it is lipid soluble, i.e. loves these lipids that we have here, will not be able to simply diffuse through. So if you are small and you are lipid soluble, you will be able to get through that phospholipid bilayer. However, if you are charged like a sodium ion, you will not be able to get through this phospholipid bilayer. Water is a special case because even though it is slightly charged, it's only slightly charged, and so it can actually sneak through because it is very, very small, these phospholipid bilayers. You'll notice that we also have some purple items here. These are the cholesterol. That sits between the fatty acid tails, and again it's a fatty substance. It helps to regulate the fluidity of the plasma membrane. So the other component then of this plasma membrane that makes up this fluid mosaic model are the proteins, and we have here intrinsic proteins. They're actually making up a channel protein, and they have a carbohydrate attached, so this makes this a intrinsic protein that's actually a glyco, meaning the carbohydrate part, protein. There's also things like extrinsic proteins, and they can sit on either side of the membrane, so you might have one actually down here, but notice they do not span the phospholipid bilayer, they only go on one side of the phospholipid bilayer, so those are not used in transport through the membrane, the plasma membrane. So how big is it then? Well, basically from here to here is 7 nanometers, so a very short distance indeed, which is obviously again important in the rate of diffusion, because anything wanting to go from the outside to the inside will need a short distance for diffusion. Plasma membrane has many different roles. If we take the glycoproteins, which we've already talked about, they can act as signalling molecules, so they can have work as receptors to bind to complementary shaped molecules such as hormones. They will have a specific shape, so that means that they will only bind to one type of molecule. They can also act as channel proteins, so allow small polar molecules to diffuse by facilitated diffusion through them. They can also be carrier proteins, and in that way, if you have a carrier protein, it will have a shape that is complementary to the shape of the molecule. The molecule may bind, and then you will require, if it's active transport, ATP to also bind. That will cause the shape to change and allow this molecule to be deposited on the opposite side. In facilitated diffusion, when the molecule binds, it will naturally cause the protein to change shape, and that's why you don't need the ATP in facilitated diffusion. But you will need a concentration gradient if you're going from this direction over here to down here. Okay, so we have, to summarise then, fluid mosaic model made up of a phospholipid bilayer. The fluidity is regulated by this molecule here, the cholesterol. You have extrinsic proteins which are only on one side of the plasma membrane. You have intrinsic glycoproteins that can act as receptors for hormone molecules, channel proteins, carrier proteins. They also act as antigens and also involved in recognition of the cell as self. You can also get proteins that act for the purpose of binding to the basement membrane, and that allows cells to bind together to form tissues. Now if we move on to the glycolipids, they have similar roles to the glycoproteins in that they can act in cell signaling, so they can act as receptors, they can act as antigens, they can have roles in communication with other cells by binding to molecules and then triggering a response on the inside of the cell. These extrinsic proteins on the inside of this plasma membrane can also be found on membranes inside the cell. So if you imagine a mitochondrion, it has a highly enfolded inner membrane which is called the cristae, and on this membrane you can find extrinsic proteins that are involved in aerobic respiration. When you're talking about the roles of membranes within cells, don't forget to mention all of the membranes involved in your organelles. So you have the mitochondrial membrane, which can control things moving in and out of the mitochondria. It's a site for metabolic reactions. There are membranes that hold onto the ribosomes, and they will be found on the rough endoplasmic reticulum. You have the Golgi, which can allow vesicles to be formed and processing and packaging. The vesicles are important because they allow transport of the substances that they contain around the cell. And you've also got the nuclear envelope, which is another membrane. It contains pores to allow mRNA to leave the cell, the nucleus, sorry, not the cell, to leave the nucleus, and as that leaves the nucleus, then protein synthesis can occur using that mRNA as an instruction. The nuclear pores are also very small, and so that means that contents within the nucleus will also be kept within the nucleus. So larger molecules like DNA will be kept inside. Okay, so that's basically your plasma membrane and the components of the plasma membrane. Good luck!